Hello, this is Dr. Sujata Magdum, Assistant Professor in Zoology, KPHM College, Nashik. Today, I'll be dealing with biological chemistry for third year BSc students, and I'll be talking about the biological buffer, which is the last topic from first chapter, that is Introduction to Biological Chemistry. In this chapter, we started with the different types of bonds like the covalent bond, non-covalent bond, hydrophobic, hydrophilic, and van der Waals forces. And we also studied the significance of these bonds in keeping the three-dimensional structures of biomolecule. You know that these biomolecules, they are freely suspended into the bodily fluid, and there is an interaction of these biomolecules with the help of enzymes to bring about the metabolic action. It means that whatever bodily fluids are present, its pH has to be maintained in spite of there is addition of hydrogen ions or depletion of hydrogen ions. In either of the cases, if the pH is not maintained, then probably the process of metabolism will come to an end and it will become lethal to the living system. And therefore, it is very essential that the bodily fluid should be maintaining its pH. In other words, we can say that there are large number of biological buffers which sustain life only because they have the ability to keep the pH unchanged, and therefore it act as a buffer, or in other sense we can say they are the biological buffer. In today's lecture, we'll be understanding the different types of biological buffers which are present into the system. Now these are mainly of three types, and they can be seen present into three different regions of the body, that is the extracellular region, intracellular region, and into the RBC, that is into the cytoplasm of the erythrocytes or RBC. Extracellular region, you all know, is the region which is surrounding to the cells or the tissue, and in this region there is presence of bicarbonate or the protein buffers, whereas within the cells or different uh, cells within the tissue, you can see presence of phosphate buffer as well as protein buffer. And in case of erythrocyte, even though it is a cell and it contains the cytoplasm similar to that of the remaining bodily cell, there is slight difference wherein you can see presence of hemoglobin, and therefore we can say that the erythrocytes, they show presence of hemoglobin buffer. So now we'll be understanding in detail the significance and composition of each type of biological buffer. To begin with, the first is bicarbonate or carbonic acid buffer system. Now this is very important buffer of the plasma, in which bicarbonate or carbonic acid buffer system is present. Here, the ratio of base to acid, that is the carbonic acid to the carbonic acid to bicarbonate in the plasma, is under the physiological conditions, and the ratio is always seen to be 20:1. The buffer system is more complex than others because the carbonic acid is formed by carbon dioxide, which is seen present into the dissolved state and is produced due to the metabolism which is occurring into the cells, and it gets diffused into the plasma. You know that whatever food is taken inside, it undergoes the process called as oxidation, and this oxidation leads to formation of large number of carbon dioxide. Now this carbon dioxide, because of difference in the partial pressure between the plasma and the cell, undergoes the process called as diffusion, which is essential to bring about its transportation, and once it is diffused, it undergoes conversion into carbonic acid in presence of an enzyme which is called as carbonic hydrogenase enzyme. Now carbon dioxide plus water, it gives rise to formation of carbonic acid, which has the tendency to once again split into the bicarbonates and the hydrogen ions. Now this is a reversible reaction which is seen occurring in the plasma, which is very slow, but in case of erythrocyte the reaction happens to be very fast because of presence of enzyme which is called as carbonic anhydrase enzyme. Now carbonic acid and bicarbonate, which is seen into the ratio as 20:1. Here the pH, as I said, is the physiological pH, and that is 7.4. When hydrogen ion concentration increases in the plasma, then the bicarbonate ion binds to hydrogen ion, forming the carbonic acid. Now this carbonic acid has the tendency to get converted into carbon dioxide and water, and this carbon dioxide which is formed, it is released into the atmosphere by the process called as exhalation, or you can say during the process of giving out of carbon dioxide to the environment through the respiratory tract, and that is called as exhalation. Now the second type of buffer is the protein buffer. In case of proteins, you know that there are ionizable groups present which are R group. Some of the examples are the carbonic acid group, which is seen present into aspartate or glutamate. Then amine group, which is seen present into the lysine, arginine and the histidine. All these are the ionizable R group, or sometimes the N terminal, which contains the alpha amine group of amino acids, are all these structures. They are responsible for the buffering in case of protein buffers. Protein, especially albumin, accounts for 95% of non-bicarbonate buffer value of the plasma. Buffering effect of protein is low in plasma. This protein is more effective buffer in intracellular medium. The most important buffer group of protein in physiological pH range are the imidazole group of histidine, which has a pK value of 6.5. Each albumin molecule contains 16 histidines. And now we'll come to the third buffer, that is the hemoglobin buffer. Hemoglobin buffer, as you know, is seen present into the erythrocytes of the RBC, erythrocytes of the blood, or you can say RBCs of the blood. Now this hemoglobin is a protein which carries oxygen to the tissue and carbon dioxide from the tissue to the lungs, and is a very effective buffer. The most important buffer group of hemoglobin are histidine. Each globulin chain, it contains 9 histidine molecules. 95% of carbon dioxide which is released from the tissue to plasma is diffused into the erythrocytes, and in erythrocyte, due to presence of carbonic anhydrase, which constitutes H2CO3, that is the carbonic acid from carbon dioxide and water, and then it has the ability to split once again into the bicarbonate, and the hydrogen ions are released by the process of ionization of carbonic acid. Now here is the reaction which is seen occurring into the erythrocytes, and in case of erythrocyte, as I mentioned here, that it contains large quantity of water for the process of carrying of CO2, and in doing so it undergoes the reaction wherein the forward reaction will tend to result into formation of carbonic acid, whereas the backward reaction will tend to result into formation of carbon dioxide and water. Now, when this condition occurs, whenever there is enhancement of H plus ion, or when there is tendency to have more accumulation of the H plus ions into the erythrocyte, it immediately binds with the bicarbonates which are present into the erythrocytes and results into formation of carbonic acid, which once again splits into water and carbon dioxide, right? So it is an interplay between the uh, increase or decrease of H plus ions to show the action of hemoglobin to get associated with carbon dioxide so that there is formation of carbohemoglobin and leads to the process of transportation. Now this once again I'll explain you that whatever released protons which takes part in the formation of salt bridges between the globulin chain of hemoglobin leads to change in the conformation of hemoglobin molecule in the capillaries, and then the binding of protons to the carbon dioxide is conversely related to binding of oxygen. In tissue capillaries, protons and carbon dioxide binding decreases the oxygen binding capacity of hemoglobin so that oxygen will be released from the hemoglobin, and this effect of, pH and carbon dioxide concentration on binding and release of oxygen by hemoglobin into the RBC is called as the Bohr's effect. Because of accumulation of large amount of bicarbonate formed by ionization of carbonic acid within the RBCs or erythrocyte, there is a concentration gradient of bicarbonates between the plasma and erythrocytes. And in that case, the bicarbonate ion rapidly move from erythrocytes to the plasma, and at the same time the chloride ions move from the plasma to the erythrocyte to provide an electrochemical balance. Now this movement of chloride from plasma to erythrocyte is referred as a chloride shift. All those phenomena which occur in the capillaries of peripheral erythrocytes conversely change in the capillaries of the lungs. That means there is always a reversible process which is seen occurring between the capillaries which are seen in the peripheral region of the body to that of the capillaries which are seen nearby to the lungs or within the lungs. When hemoglobin reacts with when the hemoglobin reaches to the lungs, the high oxygen concentration in the lungs promotes the binding of oxygen and release of protons from the broken salt bridges, and this proton will get associated with the bicarbonates, and therefore there is formation of a carbonic acid. Water and carbon dioxide formed by reaction catalyzes is I should say the water and carbon dioxide form in the reaction will be catalyzed by the enzyme which is called as carbonic anhydrase enzyme. This phenomena is called as the Haldane effect, wherein water and carbon dioxides are excreted to the atmosphere by the process called as respiration. Now the next buffer into the discussion is phosphate buffer. Phosphate buffer is most effective in intracellular medium, especially in the kidneys. Phosphoric acid has three ionization steps, that is ionization step wherein H2PO4 and H plus ions are released, and here the pKa value is 1.9. Then the second level is H2PO4. It gets converted into HPO4, 2 minus and H plus, and here in this state of reaction the pKa value will be 6.8. HPO4 2 minus which is formed, once again it has the ability to get converted into PO4 and H plus, and this is the final step wherein the pKa value happens to be 12.4. Among all these three ionization steps, H2PO4 to HPO4 is a good buffer because its pKa value is 6.8, which is very much close to the physiological pH, that is 7.4, and at this the ratio is always equal to 4. Phosphate buffer system is not effective in plasma because phosphate ion concentrations in the plasma is very less. However, it is very important in the excretion of acids in the urine. Hydrogen secreted into the tubular lumen by sodium-potassium exchanger react with HPO4 to form H2PO4, which I have just showed to you into the previous slide. Some organic phosphates like 2,3-diphosphoglycerate in erythrocyte has also buffering capacity. Now, this is a glycolytic metabolite which is formed and it plays an important role in buffering capacity. With this, we come to the end of first chapter, that is Introduction to Biological Molecules. Thank you. Thank you so much.